3B.2.4 The limbic system. At the border, limbus, between the brain's older parts and the cerebral hemispheres, the two halves of the brain, is the limbic system, figure 3.17. We will see in Unit 7a how one limbic system component, the hippocampus, processes memory. If animals or humans lose their hippocampus to surgery or injury, they become unable to process new memories of facts and episodes. For now, let's look at the limbic system's links to emotions such as fear and anger, and to basic motives such as those for food and sex. The amygdala. In the limbic system, two lima bean-sized neural clusters, the amygdala, influence aggression and fear. Figure 3.18. In 1939, psychologist Heinrich Klüver and neurosurgeon Paul Bucy surgically lesioned the part of a rhesus monkey's brain that included the amygdala. The result? The normally ill-tempered monkey turned into the most mellow of creatures. Poke it, pinch it, do virtually anything that normally would trigger a ferocious response, and still the animal remained placid. In later studies with other wild animals, including the lynx, wolverine, and wild rat, researchers noted the same effect. What then might happen if we electrically stimulated the amygdala in a normally placid domestic animal, such as a cat? Do so in one spot, and the cat prepares to attack, hissing with its back arched, its pupils dilated, its hair on end. Move the electrode only slightly within the amygdala, cage the cat with a small mouse, and now it cowers in terror. These experiments confirm the amygdala's role in rage and fear, including the perception of these emotions and the processing of emotional memories. Anderson and Phelps 2000, Poremba and Gabriel 2001. Still, we must be careful. The brain is not neatly organized into structures that correspond to our categories of behavior. Aggressive and fearful behavior involves neural activity in many brain levels. Even within the limbic system, stimulating structures other than the amygdala can evoke such behavior. If you charge your cell phone's dead battery, you can activate the phone and make a call. Yet the battery is merely one link in an integrated system that makes a cell phone work. The hypothalamus. Just below hypo, the thalamus, is the hypothalamus, figure 3.19. An important link in the chain of command governing bodily maintenance. Some neural clusters in the hypothalamus influence hunger, others regulate thirst, body temperature, and sexual behavior. The hypothalamus both monitors blood chemistry and takes orders from other parts of the brain. For example, thinking about sex in your brain's cerebral cortex can stimulate your hypothalamus to secrete hormones. These hormones, in turn, trigger the adjacent master gland, the pituitary, see figure 3.17, to influence hormones released by other glands. As we saw in Unit 3a, we again see here the interplay between the nervous and endocrine systems. The brain influences the endocrine system, which in turn influences the brain. If you were designing a robot vehicle to walk into the future and survive, you'd wire it up so that behavior that ensured the survival of the self or the species, like sex and eating, would be naturally reinforcing, as Candace Pert said in 1986. A remarkable discovery about the hypothalamus illustrates how progress in science often occurs when curious, open-minded investigators make an unexpected observation. Two young McGill University neuropsychologists, James Olds and Peter Milner, in 1954, were trying to implant an electrode in a rat's reticular formation when they made a magnificent mistake. They incorrectly placed the electrode in what they later discovered was a region of the rat's hypothalamus. Olds, 1975. Curiously, as if seeking more stimulation, the rat kept returning to the location where it had been stimulated by this misplaced electrode. On discovering their mistake, Olds and Milner alertly realized they had stumbled upon a brain center that provides a pleasurable reward. In a meticulous series of experiments, Olds, 1958, went on to locate other pleasure centers, as he called them. What the rats actually experience only they know, and they aren't telling. Rather than attribute human feelings to rats, today's scientists refer to reward centers, not pleasure centers. When allowed to press pedals to trigger their own stimulation in these areas, rats would sometimes do so at a feverish pace, up to 7,000 times per hour, until they drop from exhaustion. Moreover, to get the stimulation, they would even cross an electrified floor that a starving rat would not cross to reach food. Figure 3.20. Similar reward centers in or near the hypothalamus were later discovered in many other species, including goldfish, dolphins, and monkeys. In fact, animal research has revealed both a general reward system that triggers the release of the neurotransmitter dopamine and specific centers associated with the pleasures of eating, drinking, and sex. Animals, it seems, come equipped with built-in systems that reward activities essential to survival. Experimenters have found new ways of using limbic system stimulation to control animals' actions. By using brain stimulation to reward rats for turning left or right, Sanjeev Talwar and his colleagues, 2002, trained previously caged rats to navigate natural environments. Figure 3.21. By pressing buttons on a laptop, the researchers can direct a rat, which carries a receiver, power source, and video camera on a backpack, to turn on cue, climb trees, scurry along branches, and turn around and come back down. Their work suggests future applications in search and rescue operations. Do we humans also have limbic centers for pleasure? Indeed we do. To calm violent patients, one neurosurgeon implanted electrodes in such areas. Stimulated patients reported mild pleasure. However, unlike Olds rats, they were not driven to a frenzy. Deutsch, 1972, Hooper and Teresi, 1986. Some researchers believe that addictive disorders such as alcohol dependence, drug abuse, and binge eating may stem from a reward deficiency syndrome, a genetically disposed deficiency in the natural brain systems for pleasure and well-being that leads people to crave whatever provides that missing pleasure or relieves negative feelings. Bloom et al., 1996. Figure 3.22 locates the brain areas discussed in this unit, including the cerebral cortex, our next topic. Section 3b.3, the cerebral cortex. What functions are served by the various cerebral cortex regions? Older brain networks sustain basic life functions and enable memory, emotions, and basic drives. Newer neural networks within the cerebrum, the two large hemispheres that contribute 85% of the brain's weight, form specialized work teams that enable our perceiving, thinking, and speaking. Covering those hemispheres, like bark on a tree, is the cerebral cortex, a thin surface layer of interconnected neural cells. It is your brain's thinking crown, your body's ultimate control and information processing center. As we move up the ladder of animal life, the cerebral cortex expands, tight genetic controls relax, and the organism's adaptability increases. Frogs and other amphibians with a small cortex operate extensively on pre-programmed genetic instructions. The larger cortex of mammals offers increased capacities for learning and thinking, enabling them to be more adaptable. What makes us distinctly human mostly arises from the complex functions of our cerebral cortex. Section 3b.3.1, Structure of the Cortex. If you opened a human skull exposing the brain, you would see a wrinkled organ shaped somewhat like the meat of an oversized walnut. Without these wrinkles, a flattened cerebral cortex would require triple the area, roughly that of a very large pizza. The brain's ballooning left and right hemispheres are filled mainly with axons connecting the cortex to the brain's other regions. The cerebral cortex, that thin surface layer, contains some 20 to 23 billion nerve cells and 300 trillion synaptic connections, to Korten Myers, 2005. Being human takes a lot of nerve. Supporting these billions of nerve cells are 9 times as many spidery glial cells, glue cells. Neurons are like queen bees. On their own, they cannot feed or sheathe themselves. Glial cells are worker bees. They provide nutrients and insulating myelin, guide neural connections, and mop up ions and neurotransmitters. Glia may also play a role in learning and thinking. By chatting with neurons, they may participate in information transmission and memory. Miller, 2005. Moving up the ladder of animal life, the proportion of glia to neurons increases. A recent post-mortem analysis of Einstein's brain did not find more or larger than usual neurons, but it did reveal a much greater concentration of glial cells than found in an average Albert's head. Fields, 2004. Stepping back to consider the whole cortex, each hemisphere is divided into 4 lobes, geographic subdivisions separated by prominent fissures or folds, figure 3.23. Starting at the front of your brain and moving over the top, there are the frontal lobes, behind your forehead, the parietal lobes, at the top and to the rear, and the occipital lobes, at the back of your head. Reversing direction and moving forward just above your ears, you find the temporal lobes. Each of these 4 lobes carries out many functions, and many functions require the interplay of several lobes. Section 3B.3.2 Functions of the Cortex. More than a century ago, autopsies of people who had been partially paralyzed or speechless revealed damaged cortical areas. But this rather crude evidence did not convince researchers that specific parts of the cortex perform specific complex functions. After all, if control of speech and movement were diffused across the cortex, damage to almost any area might produce the same effect. A television with its power cord cut would go dead, but we would be fooling ourselves if we thought we had localized the picture in the cord. Motor functions. Scientists had better luck in localizing simpler brain functions. For example, in 1870, when German physicians Gustav Fritsch and Eduard Hitzig applied mild electrical stimulation to parts of a dog's cortex, they made an important discovery. They could make parts of its body move. The effects were selective. Stimulation caused movement only when applied to an arch-shaped region at the back of the frontal lobe, running roughly ear to ear across the top of the brain. Moreover, stimulating parts of this region in the left or right hemisphere cause movements of specific body parts on the opposite side of the body. Fritsch and Hitzig had discovered what is now called the motor cortex. Figure 3.24. Demonstration. Try moving your right hand in a circular motion as if polishing a table. Now start your right foot doing the same motion synchronized with the hand. Now reverse the foot motion but not the hand. Tough, huh? But easier if you try moving the left foot opposite to the right hand. The left and right limbs are controlled by opposite sides of the brain, so their opposed activities interfere less with each other. Mapping the motor cortex. Lucky for brain surgeons and their patients, the brain has no sensory receptors. Knowing this, Ottfried Forster and Wilder Penfield were able to map the motor cortex in hundreds of wide-awake patients by stimulating different cortical areas and observing the body's responses. They discovered that body areas requiring precise control, such as the fingers and mouth, occupied the greatest amount of cortical space. Spanish neuroscientist José Delgado repeatedly demonstrated the mechanics of motor behavior. In one human patient, he stimulated a spot on the left motor cortex that triggered the right hand to make a fist. Asked to keep the fingers open during the next stimulation, the patient, whose fingers closed despite his best efforts, remarked, "I guess, doctor, that your electricity is stronger than my will." Delgado, 1969, page 114. More recently, scientists have been able to predict a monkey's arm motion a tenth of a second before it moves by repeatedly measuring motor cortex activity preceding specific arm movement. Gibbs, 1996. Such findings, some researchers believe, have opened the door to a new generation of prosthetics, artificial body part replacements. Neural prosthetics. By similarly eavesdropping on the brain, could we enable someone, perhaps a paralyzed person, to move a robotic limb or command a cursor to